The overall goal of the following experiment is to generate well-replicated and highly resolved wood anatomical datasets to study the effects of time and environmental factors on tree and shrub growth. This is achieved by using a non-Newtonian fluid to stabilize the plant cells during microtome sectioning, allowing a detailed microscopic analysis of the samples. As a second step, the microsections are double stained to facilitate the differentiation of the lignified and unlignified cells, and then digital images are taken of the samples. Ultimately, automated cell-based analyses of the plant growth and related time series analyses can be performed to allow the evaluation of the wood anatomical parameters of interest.
The main advantage of this technique over existing methods like sanding the sample's core is that with our techniques the cells are not filled with sawdust, resulting in much clearer quality samples for image analysis.
Further, adding a non-Newtonian fluid to the cells during sectioning allows avoidance of the time-consuming technique of embedding the sample in paraffin.
The new microtomes you will see in the video were developed in our laboratory in cooperation with Sandro Lucinetti of the Schenkung Douglas in Zurich. They stand out due to a high stability of the guidance, which allows the cutting of big and dense specimens.
For core sampling of tree stems, begin by placing a sharpened increment core perpendicular to the growing axis of the tree stem. Next, turn in the core and use an extractor to remove the resulting core sample from the tree. Label the core with a specific ID marked in soft pencil on the core surface and repeat the procedure.